Dear students, welcome you all in the virtual process of learning. Myself, Dr. Deepika, on the behalf of School of Medical and Allied Sciences, Calcutta University, welcome you all. Today, we learn about antihistamine drugs. Histamine is a biomolecular precursor which is responsible for various allergic reactions. In today's lecture, we will understand the different drugs and approaches used for the treatment of allergy. Before we start this lecture, the student must be aware about the term allergy, which is a immune system response to a foreign substance that not typically harmful to your body. These foreign substances are known as allergens and they can include in the certain foods, pollens and the pet dander. Histamine. Histamine is ingested via the food and stored in nearly all tissues of the body. And how it is related to allergy, you must be aware about that. In today's lecture, the student will learn the different form of the histamine and the receptor binding pattern and how the substitution affect the agonist activity, the chemical classification and the SAR of H1 blockers, differentiate the receptor binding patterns and the structural feature needed between the agonist and the antagonist, different approaches in getting non-CNS active and non-sedating antihistamine drug. Introduction of histamine. Histamine, chemically known as 2,4-imidazole ethylene amine, and it is an autocoid. Autocoids means the substance which acts similarly to a local hormone near its site of synthesis. Histamine is also ingested via food and stored in nearly all tissue of the body. It is an endogenous substance synthesized, stored and released in the mast cell which are present in the skin, GIT and the respiratory tract. It is also found in the basophils that are present in the blood and some neurons in the CNS. When histamine is released, it plays a role in a ton of different action. Takes all keep your organ functioning and body working healthy. The functions like smooth muscle tissue of the lungs, uterus, and stomach contraction, dilating the blood vessels and stimulating the gastric acid secretion in the stomach, and also accelerate your heart rate. Synthesis of histamine. Histamine is synthesized in the cytoplasmic granule of its principal storage cell, that are the mast cell and basophil, from the naturally occurring amino acid, that is S-histidine, via catalysis of pyridoxal phosphate dependent histidine decarboxylase, which converts histidine into histamine. Histamine receptors. Four different histamine receptors have been categorized and are designated as H1 to H4, all of which are G protein coupled receptors. H1 receptor is majorly located on the smooth muscles, endothelial cell, and the nerve ending. The major biological effects are acute allergic responses, vasodilation, contraction of most smooth muscles except the blood vessels, means they cause the contraction in the smooth muscles. H2 receptors basically present in the gastric parietal cell or the gastric mucosa, cardiac muscle cells, mast cell and the brain. They are responsible for the stimulation of gastric secretion. S3 central nervous system. They are present in the CNS, presynaptic photoreceptor and the heteroreceptor. They are modulating the neurotransmission. H4 receptor. They are located in the intestinal tissue, spleen, thymus and the immune active cell such as T cell, neutrophils and eosinophils. They regulate the immunity. Antihistamines drugs are classified into two categories, H1 receptor antagonist and H2 receptor antagonist. In today's lecture, we will focus on the H1 receptor antagonist. The term antihistamine historically has referred to the drug that antagonize the action of histamine at the H1 receptor. H1 antagonists are now commonly subdivided into two broad groups, the first generation or the classical antihistamine and the second generation also known as the non-sedating antihistamine. The first generation of H1 antihistamine have sedative effect due to effect on the H1 receptor in the brain. Structurally, this effect is linked to their high lipophilicity, induce the blood-brain barrier penetration, and also they are the poor substrate for the brain endothelial weak glycoprotein efflux pump. Thus can't exist the brain once they enter. The second generation are more selective for H1 and do not penetrate the brain and thus have no sedation or cardiotoxicity. Mechanism of action. They bind and stabilize the inactive form of H1 receptor onto which drugs capable of binding. Classification of H1 receptor antagonists. The first class is amino-alkyl ether. Example of this category are diphenhydramine, bromodiphenylhydramine hydrochloride, dimenhydrinate, doxylamine, and the diphenpyramine hydrochloride. This is the structure of diphenhydramine. As you see in this structure, it's containing amino-alkyl ether in their structure. Second class is ethylene diamine. The drugs comes under this category are mepyramine maleate, triphenamine hydrochloride, thongylamine hydrochloride, and the jolamines. Look at the structure of triphenamines. It contains two ethylene and two amines. That's why they are known as ethylene diamine in their structure. All drugs comes under this category having this common group in their structure. The third category is thiophene derivative. Examples are the methapyrrolene, methaphenylene, and the thylene diamine chloride and the chlorothene citrate. The fourth category is alkyl amine derivative. Example is chlorpheniramine maleate. As you see in the example, the heteroatom is changed with the carbon and that is known as the alkyl amine. Now continue the classification of H1 receptor antagonist. The next class is cyclic basic chain analogs. Means they containing nitrogen atom in the chain. Example are imidazoline derivative. Drugs under this category are antazoline, piperazine derivative, cyclazine hydrochloride, chlorcyclazine hydrochloride, maclazine hydrochloride, buclazine hydrochloride. Look at the structure of cyclazines. It containing piperazine ring in structure. So drug comes under this category containing the piperazine ring in their structure. Piperidine derivative, phenylidine tartrate. The next category is phenothiazine derivative. Example of this category are promethazine hydrochloride, promethazine ticuclate, trimerazine tartrate, methadilazine hydrochloride. Look at the structure. It containing the phenyl ring and phenylthiazine ring in their structure. Second generation non-sedating antihistamine are the terfenadine, estamizole, lortadine, aclivestine, and fexofenadine. The drugs which comes under miscellaneous categories are phenidamine, propylidine, and the cycloheptadine. Now move to the SAR of H1 antagonist. The general structure of H1 antagonist consists of two diaryl moieties, one connecting atom, a spacer which consists of 2 to 5 carbon chain, and a substituted amines. The diaryl substitution pattern is present in both first and second generation antihistamine and is essential for significant H1 receptor affinity. Most H1 antagonists contain substitution in one of the aryl ring is usually benzene and the other containing the pyridine ring, and these influence the antihistamine potency as well as the biodeposition. In many of the first generation antihistamine, the terminal nitrogen atom is a simply dimethylamino moiety. In all the cases, amino moiety is basic with dissociation constant ranging from 8.5 to 10 and thus presumed to be protonated when bound on the receptor. X is a connective atom of carbon, oxygen and nitrogen. The X connecting moiety of typical H1 antagonist may be a saturated carbon-oxygen moiety or simply a carbon or nitrogen atom. This group, along with the carbon chain, appear to serve primarily as a spacer group for the key pharmacophoric moieties. The nitrogen or the amine may also be a part of a heterocyclic structure as illustrated by the piperazine and the some propylamine derivative like the pyrrolidine and piperidine and the some phenothiazine ring and the dibenzocycloheptene as present in the second generation H1 antagonist. Continue with SAR of H1 antagonist. Many of the antihistamine containing a carbon atom in the connective moiety are chiral in nature and exhibit the stereoselective receptor binding. For example, in the phenyramine series and the carbinoxamine, this atom is chiral and in vitro analysis indicate that those enantiomer with the N configuration have the higher H1 receptor affinity as compared to the R configuration. So, an enantiomer having the highest and optimum activity. The methyl group and the connective atom result in a distance between the central point of the diaryl system and the terminal nitrogen atom in the extended conformation of the antihistamine ranging from 5 to 6 angstrom, that is a special group. In some series, branching of the carbon chain result in the reduction of antihistaminic activity. So the optimum distance between diaryl and the substituent amine is 5 to 6 angstrom. However, a stereoselective H1 receptor antagonist typically is not observed when the chirality exists at this site. Also in those compounds which possess an asymmetrically substituted unsaturated carbon chain, for example, pyrobutamine and the tropylidine, one geometrical isomers typically display the high receptor affinity than the other. Summary of today's lecture. Histamine, chemically known as 2,4-imidazole ethylamine, and it is an autocoid. Autocoids are generally the local hormone which is synthesized its near site. It is an endogenous substance synthesized, stored and released in mast cell, basophils that are present in the blood and some neurons in the CNS. Histamine is synthesized from naturally occurring amino acid as histidine via catalysis of pyridoxal phosphate dependent enzyme, that is histidine decarboxylase. 4 different histamine receptors have been characterized and are designated as H1 to H4, all of which are H1 protein coupled receptors. The H1 antagonists are now commonly subdivided into two broad groups, the first generation or the classical antihistamine and the second generation or non-sedating antihistamine. SAR is important to evaluate the overall physiochemical properties of the medicine. In H1 antagonist, X connecting moieties and the nature of substituents in the alkyl side chain or the terminal nitrogen among the various groups account for the difference observed in the antihistaminic potency as well as pharmacological biodisposition and the adverse drug profile. Here are some practice questions from the today lecture that are important for the exam point of view. What is the difference between first generation and second generation antihistamine? the basic structural requirement for the antihistamine, and what isosteric replacement reinforce the activity, which aromatic substitution increase the activity and which substitution decrease the activity. Basically, you have to explain the SAR of H1 antagonist. What are the different class of antihistamine? You have to elaborate all the classes. Details are of antihistamine, what is the basic receptor binding and what are the mode of antihistamine, is the antihistamine binding site chiral, and which nitrogen atom of antihistamine structure is more basic and thus cause the charge interaction with the receptor. Yes students, if you want more information regarding this lecture, you can refer to these books. We will meet in the next lecture. Till then, Namaste.